Hello students, today we are going to discuss about dicot stem. In our previous lecture we have already discussed about the anatomy of root. So let's start. First of all we will discuss about epidermis. Epidermis is covered with a thin layer of cuticle and may have trichomes and stomata. Then cortex is made up of multiple layers of cells including hypodermis, middle layer of parenchyma cell and innermost layer called endodermis. Endodermis cells are rich in starch grains and are called the starch sheath. Pericycle is present on the inner side of endodermis. Layers of radially placed parenchyma between the vascular bundles are called medullary rays. And we see the structure, a large number of vascular bundles are arranged in a ring. Basically, the structure, each vascular bundle is conjoint open, protoxylem is endarch. Open, that means between xylem and phloem there is a cells of cambium is present. Monocotyledon stem, यानी monocot stem में क्या difference है? The hypodermis is made up of sclerenchyma. Vascular bundles are conjoint close. There is no vascular cambium. There is no cambium cells. And scattered, each vascular bundle is surrounded by a sclerenchymatous bundle sheath. Phloem parenchyma is absent. Water containing cavities are present within the vascular bundles. Next is leaf and that is dorsiventral or dicot leaf. The leaf lamina of a dorsiventral leaf has three parts: epidermis, mesophyll and vascular system. The upper epidermis is called adaxial and lower one is called abaxial. More number of stomata are present on the abaxial epidermis, that is lower side. There are two types of cells in the mesophyll, palisade parenchyma and a spongy parenchyma. The palisade parenchyma is placed adaxially. The spongy parenchyma is situated below the palisade parenchyma and extends to the lower epidermis. There are numerous large spaces and air cavities between the cells of spongy parenchyma. Basically, the vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. And the difference between dicot and monocot leaf is stomata are present on both the surface of an isobilateral or monocot leaf. The mesophyll is not differentiated into palisade and spongy parenchyma. Some adaxial epidermis cells in grasses are modified into large empty cells called bulliform cells. When the bulliform cells absorb water, they become turgid. So the leaf surface is exposed. During water stress, when the bulliform cells become flaccid, the leaves curls inward to minimize water loss. Next, our topic is secondary growth, which one is most important topic of anatomy. What is secondary growth? The increase in girth of a plant body is called secondary growth. The tissues involved in secondary growth are vascular cambium and core cambium. We know that the plant increase lengthwise by the activity of meristematic tissue and increase in the girth by the activity of vascular cambium. That is secondary growth. So what is vascular cambium? In case of young stem, vascular cambium is present in patches as a single layer between the xylem and phloem. It forms a complete ring at a later stage. Activity of the cambial ring. The cambial ring becomes active and begins to cut off new cells both towards the inner side and outer sides. That is, outer side is secondary phloem and inner side is secondary xylem, which is dead tissue. These cells which are cut off towards pith mature into secondary xylem. The cells which are cut off towards periphery mature into secondary phloem. The cambium is more active on the inner side than on the outer. As a result, the amount of secondary xylem produced is more than secondary phloem. The primary and secondary phloems get gradually crushed due to the continued formation and accumulation of secondary phloem, which is the place of the storage of tannin, lignin and resins and other materials. At some places, the cambium forms a narrow band of parenchyma which passes through the secondary xylem and the secondary phloem in radial directions. These are the secondary medullary rays. We can see in the diagram the process of secondary growth. First of all, the cambium cells start divide, which is interfascicular cambium. Intrafascicular cambium start dividing and forms interfascicular cambium, then the ring complete and so on. Next our topic is our spring wood and autumn wood. Cambium is very active during the spring season but less active during the winters. Hence, during spring, a large number of xylem elements are formed having wider vessels. During winter, less xylem elements are formed having narrow vessels. The wood formed during summer is called spring wood. The wood formed during winter is called autumn wood. The two kinds of wood appear as alternate concentric ring in transverse section of a trunk of a tree. These are called annual rings and provide information about age of the tree. The reason of the formation of these rings are the activity of cambial cells. We can say the activity of cambium cells are more in spring season or the summer season, and they are less active during winter season. Next is heartwood and sapwood. In all trees, the greater part of secondary xylem is dark in color, hard and resistant to attacks by microorganisms and insects. This resin is made of dead element with highly lignified walls. This wood is called heartwood, and the heartwood gives mechanical support but doesn't conduct water. The peripheral part of the secondary xylem is lightly colored. This is known as sapwood. It helps in the conduction of water and minerals. Next is core cambium. Meristematic tissue which develops in the cortex region is called core cambium or phallogen. The phallogen cuts off cells on both sides. The outer cells differentiate to form cork or phellum, while the inner cells differentiate into secondary cortex or phalloderm. Phallogen, phellum and phalloderm are collectively called periderm. Due to activity of core cambium, pressure builds up on the remaining layers peripheral to the phallogen. These layers gradually die and fall off. Next is lenticels. A certain region, the phallogen cuts off closely arranged parenchymatous cells on the outer side instead of cork cells. These parenchymatous cells soon rupture the epidermis, forms a lens-shaped opening called lenticels. Lenticels permit the exchange of gases between the outer atmosphere and the internal tissue of the stem. Next is secondary growth in roots. The vascular cambium of the dicot root originates from the tissue located just below the phloem bundles. A portion of pericycle tissue present above the protoxylem forms a continuous wavy ring. We can say the structure of dicot stem in which the ring is continuous, while in this the ring is wavy. It gradually becomes circular. Rest of the steps are similar as in dicot stem. Secondary growth takes place in stems and roots of gymnosperm. No secondary growth occurs in monocots except some exceptions. So this is the topic of anatomy. Thank you.